Firstly, 2 grains of rice are collected in order to grind them. After grinding, we place it into a capillary tube. As you can see, it is important to put plasticine on both sides of the capillary tube. Then it is placed on a laboratory centrifuge in which the sample side of the capillary tube is placed outwards. We have to take into account that the speed and time depends on the type of the sample. For example, in rice samples, it is used 5 rpm for 5 minutes. In olive samples, it is used 2 rpm for 2 minutes. And for cell samples, it is used 2 rpm for 20 seconds in order to not kill them. Lastly, we put the capillary tube in a probe in which the sample side is placed downward. Then it is inserted in the NMR system. Firstly, we should turn on the machine 30 minutes before acquisition to stabilize the temperature to 30 degrees. Then, open program PROSPA v3.56 and go to Kia NMR. The first thing to do is the wobble. Wobble is a macro which measures the amount of radio frequency energy reflected from the probe. The wobble macro simultaneously sweeps the radio frequency pulse and receiver frequency, and for each frequency step, the receiver acquires a number of points which are averaged and then displayed on the screen. So we should put run, adjust screws while running to get a V-shape in the graphic with the peak around the center frequency, then finish and save the photo. After the wobble, we have to determine the correct B1 frequency. For that, we use CPMG freq sweep, which is a spin-echo experiment in which the pulse and detection frequency is varied to find the optimal NMR resonant frequency. CPMG coding of echoes is included to enhance the SNR. The collected echo sum is Fourier transformed, and the resultant peak integral is plotted. From the maximum in the curve, we can determine the correct B1 frequency. It is important to take into account that to determine the correct B1 frequency, we should put run to test if the guessing value is actually accurate. Then, we check the highest value in the pulse frequency analysis. If the value is inside the half fit of the previous graph, we continue to measure the other macros. However, if it is not the case, wobble should be repeated. CPMG-int is used to do the T2 measurements. Firstly, we have to change B1 frequency to the value calculated in the previous step. Afterwards, we have to change the echo time and the number of echoes to allow the curve to reach 0. Also, the number of scans, which is a multiple of 4 and is highly dependent on the homogeneity of the sample. The more homogeneous, the less scans is needed. The processing tab in the CPMG-int experiment is non-standard. It provides 3 ways to analyze the data. In addition to a single exponential fit, there is a b-exponential fit, and if the inverse Laplace fit option is selected, a spectrum of exponentials will be fitted to the decay curve. T1 IR-ED is a standard inversion recovery experiment with CPMG detection added for measuring T1 in a homogeneous magnetic environment. This can result in a considerable improvement in the signal-to-noise ratio when compared to the simpler T1 IR echo experiment. It is also important to notice that the maximum delay influences the length of the plot, so it should be high enough in order to the plot reaches 0. Lastly, T1-T2 correlation implements the T1-T2 experiment. The first part of the experiment is a conventional inversion recovery pulse sequence used to encode for T1 relaxation. The second part is a CPMG sequence used to encode for T2 relaxation. The pulse sequence is executed a number of times varying the inversion recovery delay. The resultant T2 dataset can be analyzed to obtain a T1, T2 distribution plot. In this plot, T1 will be in the y-axis and T2 will be in the x-axis. Also, we use the values calculated previously, the echo time, the number of echoes, and the maximum tau. The real part of this data may be then analyzed using a two-dimensional inverse Laplace transform. For that, we have to change the steps from 90 to 200. Afterwards, we zoom in the red area of the plots and copy the values.